So today I'm going to just touch upon what for me were highlights of ASH in the lymphoid malignancies. I will not be going into myeloma or the myeloid malignancies. Without further ado, in CLL, one of the trials that was interesting was the AMPLIFY trial. In the AMPLIFY trial, patients received a control arm of chemoimmunotherapy or one of two investigational arms of acalabrutinib and venetoclax, with or without obinutuzumab. And what was found was that the acalabrutinib and venetoclax arms improved outcomes compared with chemoimmunotherapy, perhaps not very surprisingly. There was, as has been seen in other studies, increased toxicity when the obinutuzumab anti-CD20 antibody was added to acala and ven. For me, probably the winner of that trial was the acala-ven combination. So I would say this trial is of particular interest because it opens up the possibility for an FDA approval of that two-drug oral combination of acala and ven for initial therapy for chronic lymphocytic leukemia. As you know, the combination of ibrutinib and venetoclax has been more extensively studied in several trials and approved ex-US, but was not approved by the FDA in the US. And so this would open up the first BTK inhibitor plus ven combination as a potential to be approved and available for use in patients with CLL. So that I think is an important trial to be aware of. A second important CLL trial was Bruin CLL 321, which compared pirtobrutinib with investigators' choice of bendamustine and rituximab or idelalisib and rituximab in patients with relapsed or refractory CLL. And pirtobrutinib, the non-covalent BTK inhibitor, was clearly superior to investigators' choice of control arm. Interestingly, about half the patients on the study had received prior venetoclax and the other half had not. So pirtobrutinib demonstrated real activity in both groups. And so it does raise the possibility of a novel sequencing option where a patient could get a covalent BTK inhibitor frontline and then use pirtobrutinib as a second line while saving venetoclax for later. It doesn't really answer the question that that's the best way to do it, but it certainly raises that as a viable option for patients. So sort of actually probably raises more sequencing questions in CLL than it answers, but it does give that option. So that was an interesting trial. And then a third CLL trial that's not perhaps practice changing right now, but was of great interest was EPCORE CLL1. This was a trial of the bispecific antibody epcoritamab, which targets CD3 and CD20 in patients with relapsed chronic lymphocytic leukemia. It's a single-arm study with 40 patients, and again, not practice-changing at this point, but they demonstrated an overall response rate of 61% and a complete response rate of 39%, which I find to be encouraging in a heavily pretreated CLL population. The drug does cause probably more cytokine release syndrome in CLL than it does in lymphomas, and the rate of that appeared to depend on the dose and schedule that were used. So that's going to be something to look out for, but it does sort of signal that bispecific antibodies have enough activity in relapsed CLL to, certainly warrant further investigation and may eventually reach approval in the long run. So those were kind of some of the interesting ones for me in CLL. This was a big meeting for mantle cell lymphoma. Probably the biggest result was the Triangle trial, which wasn't the first time we've heard from Triangle. They reported this 2 years prior at ASH. This was a frontline comparing chemoimmunotherapy induction with or without auto transplant and with or without ibrutinib added to the initial regimen. And we had learned a couple years ago that adding ibrutinib was improving failure-free survival. What we saw at this ASH meeting was that with 2 additional years of follow-up, the same holds true. There appears to be some trends in overall survival, and it further confirmed the signal that was seen a couple years ago, namely that if ibrutinib was added to the initial therapy and the long-term maintenance, there did not appear to be any benefit to autologous stem cell transplant. in treating these patients who were all transplant eligible. So for me, this trial sort of further solidified what I suspected from 2 years ago, which is that probably when I'm seeing a transplant eligible patient with newly diagnosed mantle cell lymphoma these days, if I add a BTK inhibitor to the chemoimmunotherapy regimen, I probably can skip the transplant for most patients. Sort of a big takeaway that I think will change practice a bit. And then the question becomes, well, what BTK inhibitor do you add since ibrutinib was withdrawn from the market? We saw data on acalabrutinib we've seen that now on a couple of other trials in mantle cell lymphoma. So probably my go-to will be acalabrutinib added to chemoimmunotherapy as initial therapy for mantle cell lymphoma. Another interesting mantle cell lymphoma trial, different induction regimens. This was the ECOG-ACRIN EA4181 trial, comparing 3 different induction regimens. And this was not a long-term follow-up, but they looked primarily at the complete metabolic response rate for 3 different regimens, and those were bendamustine rituximab plus acala, bendamustine rituximab plus cytarabine and rituximab, and then bendamustine rituximab plus cytarabine rituximab with acala added to that. So three-arm trial. And the bottom line on this one, I would say, is that the addition of cytarabine to the bendamustine rituximab did not really improve outcomes and added toxicity, which argues somewhat against the routine use of that regimen with the acala added in there. And the least toxic of the regimens was benda rituximab plus acala, and it appeared to have very similar efficacy, at least in terms of response rate, as the other arms. And so in my mind, this trial raises the question as to whether this high-dose cytarabine is really necessary. I've been kind of personally questioning that myself based on a SWOG study that was reported years ago. And I think it confirms that, at least especially for community practice doctors, that I think bendamustine plus rituximab plus acalabrutinib is a reasonable induction regimen, even in transplant eligible patients with newly diagnosed mantle cell lymphoma. So that was kind of my combined takeaway from some of the initial mantle cell lymphoma trials. There were a lot of reports of chemo-free regimens being tested in mantle cell lymphoma. And clearly, the field of mantle cell lymphoma appears to be moving in the same direction as the direction that CLL has already moved in, which is elimination of chemotherapy in the front line. I'm not sure that that's ready to be declared a standard yet, but there were trials looking at that and continue to be trials looking at that. And so that may be the way we're headed in mantle cell lymphoma. And then finally, I will touch on InMind, which was a relapsed follicular lymphoma study comparing a control arm of lenalidomide plus rituximab, which is a standard regimen in relapsed FL, versus the investigational arm of the same plus the CD19 antibody tafasitamab. The addition of tafasitamab nearly doubled the median progression-free survival. It improved other endpoints like duration of response, time to next treatment. There was no overall survival difference yet, but there actually did appear to be somewhat of a trend favoring tafasitamab, and the differences in the adverse events were fairly modest. So this is a strong signal that adding tafasitamab to the R-squared standard regimen in relapsed FL really provides the potential for a benefit for patients. And so that may become a new standard once it goes through regulatory approval and NCCN evaluation, and perhaps with some longer follow-up. So that was of great interest in relapsed FL. So those are some of the highlights for me. I certainly can't cover everything, but some of the highlights for me in the lymphoid malignancies at ASH.